The immune system has an innate component, and in the innate component, your experience doesn't matter. It will work just as well regardless of where you were born, regardless of where you went on vacation, whether you were vaccinated. But the reason that we can live so long is because we have a third line of defense, an adaptive or an acquired immune response. Now, this adaptive or acquired immune response is so complex that we split it into two groups. The lymphocytes are the primary cells which govern the adaptive or acquired immune response. All lymphocytes develop in the bone marrow. We stimulate those pluripotent hematopoietic stem cells to produce lymphocytes. But then a question then arises, where do these lymphocytes then complete their development? Those which complete their development in the bone marrow, they become B lymphocytes, and they are capable of making antibodies which can be secreted. Those which go to the thymus, they then become T lymphocytes. They don't have antibodies, they have T cell receptors, which are similar. They're not quite as variable. There are only millions of combinations of these, as opposed to billions of combinations of these. And T cell receptors cannot be secreted, so they do not have a humoral aspect which can be secreted in the body's liquids or humors. Instead, T lymphocytes must contact other cells with which they are interacting. That's why this is called the cell-mediated response. Now, B lymphocytes can go after antigens, and then T lymphocytes can go after antigens as well. However, the antigens that T lymphocytes go after, they are bound to self proteins or MHC proteins. Now, this is a bit involved, so I'm going to explain it, but let me just kind of very quickly, give an overview and then go into it in more detail. So your cells have ID badges. Just like if you were walking into a high security area, you'd be asked to show ID. That people with correct ID, they're allowed, all right, but people without correct ID, they're not allowed, and security guards would check that. So your T lymphocytes can essentially bump into a cell and say, could I see your ID? And so therefore your cells say, here's my ID, which show that I bind, that I belong to this person, I belong in this body. Having the right ID is important. As we will see, these ID badges actually bind to proteins inside the cell, or actually better said, peptides, short fragments. So that ID badge, before it came to the surface, it actually was able to bind something from the middle of the cell. So not only do you say, here's my ID, you can actually prove what's going on inside you. You can say, and here's a little piece of what is produced inside the cell for reference. That becomes very important, and I'll get to that in just a few minutes. So those cell ID badges are called the major histocompatibility complex, or MHC proteins. They are quite involved, and I'll go into them a little bit more in detail in the future when I talk about type 1 diabetes and autoimmune diseases. So this is just our introduction into it here. So what does the cell ID badge look like? Well, it's proteins that your body makes. Your body makes the ID badge. The most important part that I'll consider are these aqua parts, which are coming from the human leukocyte antigen HLA genes on chromosome 6. But there's another part, the beta-2 microglobulin coming from chromosome 15. So that's your cell ID badge. Now, as I said, it can bind a peptide from the cytoplasm, whose importance we talk about later, and that top piece, that was that. So on your chromosome 6, you have genes for a cell ID badges. Interestingly, these are the most variable genes known. So when you ask what makes your blood type genes different from the person next to you, or hair color genes different from the person next to you, the odds are there aren't that many differences. In our big human population, most people have, either the same form of the gene or only one of a couple of common variants of the gene. However, these LA genes. For this first one, there's more than 3,000 alleles known. For the second one, over 3,800 alleles known. That's a lot of variation. So your cell ID badges are very likely to be different from the person next to you. Your hair color genes, your genes for height or blood pressure, not so much. So these are very variable genes. And as a result, the cells of this person are expressing these ID badges, which look different from the cell ID badges of this person versus that person. Now, this person actually has two of the same, so their mother and father, maybe were of the same ethnic group, and so therefore had this two of the same. So there's a little less variability there. So as a result, inside your body, your cells have these cell ID badges. White blood cells can bump into cells and say, "Hey, could I see your ID?" And they learn, we'll get to this in a second, they learn to recognize self and not react against self. So your white blood cells touch this cell and say, you have self ID. You have the correct ID belonging to our body. You can pass. This other cell over here doesn't have the correct ID. And so that it would be recognized as foreign. And this is why, even if I say need a heart and I get a heart transplant, or I get a bone marrow transplant, or some sort of a transplant, even though it's a human heart and I need a human heart, the human heart and the transplant might have different ID badges. And so therefore, my immune cells would recognize it as foreign and attack it, even though I need this organ. That's why this is called the histocompatibility complex. You need to check these IDs if you're doing a transplant to try to get a donor who is most compatible to the one receiving the organ. So it's kind of like a fake ID. If the fake ID looks very different from the person who's pretending to have that identity, it doesn't work. So it would only work if someone looks reasonably similar. And so we have to screen transplanted organs for MHC proteins, which look similar. So our T lymphocytes, they have receptors. The receptors are similar to antibodies in that we shuffle the parts of them and create a great variety. That's good because there's millions of different kinds of, a gazillion different kinds of bacteria. So just like we have different types of antibodies because we shuffle antibody parts, the same thing for T cell receptors. The difference being that there are millions of different kinds of T cell receptors, and billions of kinds of antibodies, and the T cell receptors, they are not secreted from the body. Okay. Now, the key with these receptors is this. Now, this is incredibly varied, and so this is, a major component. There are others, but a neutrophil can touch a cell and say, you're a bacteria. That's bad. But this cell is a cancer cell. It's a cell of your own body. So how can you defend against cells of your own body which have gone bad? Well, the answer is those ID badges, if you recall. Those ID badges are going to grab something. See, we're grabbing something and then taking it to the cell surface. So that little yellow speck there, that's a peptide, a protein fragment from the inside of the cell. So when the T lymphocyte touches the cell and says, "Can I see some ID?" it's going to say, you have the correct ID. You're a cell of the body. Ooh, wait, but look at this yellow part. I don't like that. See, that's very valuable because even though this is a body cell, this T lymphocyte now knows there's something wrong inside. You're sick. Maybe we should kill you. So that's incredibly valuable. So I have all of these genes. Now, as I'm an embryo, my immune system is learning which ones, I tend to express. And cancer cells can express abnormal proteins they shouldn't be. So cancer cells are by definition abnormal. They're not regulating their genes as they should. They're using genes that they shouldn't. And so this might cause them to make abnormal protein. So for example, let's say sometime in the future, I get blood work done, and by the blood work, someone says, it looks like you're pregnant. You make proteins which we associate with pregnancy. Does that mean that I'm pregnant? It does not. It means that I have cancer. I shouldn't be making pregnancy proteins. If I am making pregnancy proteins, that means something's going wrong. Cells are expressing proteins that they shouldn't. These cells are quite likely cancerous, doing many things that they shouldn't. So here's an example. That then could be an abnormal peptide, not a normal protein. And so my immune system learned what's normal and now recognizes, ooh, I don't like that at all. So we could actually identify cancer cells. Your immune system obviously doesn't pick up all cancer cells, or else there wouldn't be cancer. But in HIV patients who don't have these lymphocytes, or else that they are not working, cancer is more common. So for example, Kaposi sarcoma is a cancer which most people don't develop, but AIDS patients do. Does AIDS cause cancer? It does not. But apparently this type of cancer pops up on occasion, not that infrequently. A healthy immune system recognizes these cells. You're foreign. I don't like what's going on inside your cell. I'm going to kill you. If these don't work, as they don't in HIV, now all of a sudden, here's a cancer that we can't detect. Here's another variation. This isn't a cancer cell. It's a human cell infected with a virus. Now, it's a human cell, but there's virus on the inside making virus peptides. As a result, when this immune cell says, this T lymphocyte says, can I see some ID? It sees the correct ID, but then it sees that abnormal peptide from the middle of the cell that was encoded by the virus. This immune cell says, you're part of the body, but you're sick inside. You must have a virus. I think you should be killed. And so T lymphocytes, they're looking for two things primarily. They are looking for both the cell ID badge and the peptide from inside the cell. All right. Now, there's other variations. So cytotoxic T cells could kill a fungus just because they don't like the fungus. But this is the main function of the T lymphocytes. They're looking for two things, the ID badge and the peptide, because when you put those two things together, it means that this is a body cell which is sick inside that needs to be destroyed. So T lymphocytes can offer protection against viruses and offer a protection against cancer cells that and that's kind of a unique ability of those. So T cells have these receptors which are looking for two things. One, a cell ID badge, and then an antigen bound to it. That makes them special. Now, T lymphocytes are incredibly complex and can then be subdivided into categories. This gets complex very quickly. So I just want to introduce the categories very quickly and not go into incredible detail. So for example, there are what are called Treg cells, sometimes called T suppressor cells. These Treg cells, this one, for example, one of the roles that it has is to help T cells develop. So as we're asking, are we doing more Th1 T lymphocytes, which will fight, say, bacteria, or more Th2 T lymphocytes, which will cause inflammation? These Treg cells are helping the immune system mature. And so therefore, that's important. You may recall that as a fetus, this person who is AB positive, so they have the A glycoproteins, the B glycoproteins, and the RH glycoproteins, their red blood cells are AB positive. They would potentially make antibodies which would attack yellow. They make anti-A antibodies, anti-B, and anti-RH. But during fetal development, you realize, wait a minute, I don't want to do that. That would cause an autoimmune disease. So here comes the Treg cell that says, sorry, none of you guys can be active. We're either going to kill you or you should be inactivated so that you do not cause an autoimmune disease. So one of the classes of a T lymphocyte is what's called a T suppressor cell or a Treg cell. Now, it sounds odd that we would have suppressor cells. Why would we want to suppress the immune system? Well, and that's because, I mean, quite frankly, immune cells are killers. All right, immune cells very often say they touch another cell and then ask, do I kill you? And so one of the things we don't want is cells that we react against self. We don't want inappropriate immune reactions. And so Treg cells are important for this. This cytotoxic T cell would kill this body cell because it mistakenly confuses its peptide as foreign. This T helper cell would mistakenly encourage an immune reaction, which would react against self. And so that can't be. So this cell is bad, this cell is bad. So Treg cells have to suppress immune responses. We don't want to react against self. So there's genetic disorders where this doesn't work, all right, that we're not regulating this, and this can cause autoimmune diseases where we actually attack ourselves. And one of the important places this happens is in these corpuscles of the thymus. We are helping to educate our immune system. And so Treg cells or T suppressor cells, they're important because we have to, train our immune cells. We want immune cells which will react against foreign peptides, but we don't want immune cells which will react against self, because that would then cause an autoimmune disease. And so therefore, as T lymphocytes mature, there are steps in this. There is positive selection. We want T lymphocytes which react against foreign things. And then we have negative selection. We don't want T lymphocytes which react against self. And Treg cells help in this process. So one of the kinds of T lymphocyte has a regulatory function. It helps to promote the appropriate maturation of the immune system and inhibit cells which would react against self. A second class of T lymphocyte are what are called cytotoxic T lymphocytes, or killer T cells. And what they do is they won't go up to cells and they say, could I please see some ID? And if they don't like what they see, all right, so if this cytotoxic T lymphocyte touches this cell and says, ooh, you have ID, but that's a foreign peptide, I think you're cancer, or I think you're infected with a virus, you should die. These cytotoxic T lymphocytes have two ways of killing a cell. So one, and I apologize, this tiny segment of the video works backwards. I screwed up, so my bad. What this cell can do is reminiscent of the complement proteins. This killer T cell, this cytotoxic T cell, can secrete these little yellow pieces called perforins. Now, perforins come together and form this, a channel. So if this cytotoxic T cell is touching a cell that it doesn't like, it can secrete pieces of a channel. The pieces then assemble, and once again, we have essentially stabbed the cell. So look, because this cell didn't like this virally infected cell, it made that piece, that piece, that piece, that piece. They assembled to make a pore, a hole on this cell of this virally infected cell. And now that we have a hole in the cell, we generated a pore, this cell is now going to die. So the killer T cell killed the virally infected cell, or killed the cancer cell, because it secreted perforins in pieces that assembled then on the infected cell membranes and generated a hole. Once again, extra holes are bad. Good stuff can leak out, bad stuff can leak in. You can't regulate stuff across the cell membrane. And so now the cell is dead. So cytotoxic cells can kill infected cells with virus or cancer cells by putting holes in their membrane. They secrete perforins. There is also a second way that killer T cells can work. This is very involved and involves a bit more genetics, so let me just quickly mention this. Cells have self-destruct switches. We know that cancer is a thing. We know that if we're going to develop into an embryo, sometimes we want to kill our own cells. So for example, I once had a paddle for a limb. And then I realized, wait, I don't want paddle. I don't want cells here, 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 and here. I don't want webbing. I want these cells to die. So they did. And now look at me, I don't have webbing. Think of it like your garden. Your garden has to be pruned. You want this weed to come up, you know, you want this branch to be pruned, et cetera. So a perfect garden just doesn't grow. Sometimes you have to kill things in your garden, this branch, this weed, for it to look the way it does. And so we know this. And so our cells have self-destruct switches so that we can prune them if needed, so that we can do what's called programmed cell death or apoptosis. So this is part of normal embryonic development, normal development of the brain. But cytotoxic T cells can trigger that. So a cytotoxic T cell can say, hey, you should die. You're a cancer cell. You're an infected cell. We're going to trigger the apoptosis pathway. We're going to turn on those genes, which then will cause a cell to kill itself. All right, so this is involved. It involves, among other things, releasing proteins from the mitochondria like cytochrome C. So cells have self-destruct switches. If you activate apoptosis, programmed cell death, then the cells will die. Cytotoxic T lymphocytes do this. Cytotoxic T lymphocytes, they are part of the adaptive immune response. There's also another interesting type of cell I just want to mention called NK cells, or natural killer cells. They are not quite cytotoxic T lymphocytes. They have some of the features of cytotoxic T lymphocytes, but not all. They don't mature in the thymus. They don't really recognize a cell ID badge. They just kind of count them. So they just ask, does this person make the normal amount of ID badges? And if it doesn't, it says, ooh, maybe you're infected or cancer, you should die. It's not actually recognizing the ID. It's just kind of checking that you have it, if that makes sense. So NK cells, they also can secrete perforins and kill potentially cancer cells or virally infected cells. Interestingly, they're more in the innate immune system. They're kind of halfway between a T lymphocyte of the adaptive immune system and an innate cell. So they're interesting. I just wanted to mention these are like cytotoxic T cells. So we have T lymphocytes which can kill cells which are cancerous or which are infected by virus, and then even NK cells which are similar to them. Our final type of T lymphocyte is what's called a helper T lymphocyte. Oh, and I'm sorry, just getting back to those Treg cells. Once again, autoimmune disease is when your immune system attacks itself. So I'm attacking my own joint linings as foreign. I shouldn't, all right, but now I have rheumatoid arthritis because I am. So I'm killing my own joint linings. I'm killing my own beta cells of the pancreas, which make insulin, and now I'm going to have type 1 diabetes. So that's an autoimmune disease where my body attacks itself. And the Treg cells help to prevent that. Okay. The last type of T lymphocyte is what's called a T helper cell, all right? Now, I had kind of lied to you before. It is possible for B cells to make antibodies without the help of T lymphocytes. It is possible, but that's not usually what happens. What usually happens is this. Here's a B lymphocyte. It binds to a microbe. And you ask, now, does it divide, make plasma cells, make antibodies, make memory cells? No. It just sits there and does nothing. Why? Well, the answer is because it doesn't have permission, all right? It needs permission in order to divide, all right? And the T helper cell is a permission giver. It will come up and say, oh, hello, B lymphocyte, you've bound a microbe. I'm going to give you a local hormone interleukin, which then stimulates you to then start the immune response, okay? So this B lymphocyte, it didn't go into action until the helper T lymphocyte gave it permission, all right? And so the T lymphocytes, they are needed for B lymphocytes to function for the most part. I had lied to you before when I said a cytotoxic T lymphocyte would recognize a cancer cell, make perforins, and kill it. No, that's not true. It sits there and does nothing until a T helper cell comes along and gives it permission. So this cytotoxic T cell knows that that's cancer. Is it killing it? No, it's not. Not until it has permission. So this helper T cell has to come over and give this cell permission by making local hormones called cytokines, like interleukin. So when the T helper cell makes interleukin, then this cell can then say, aha, I now have permission to act. Now, I know this sounds like an unnecessary step where B lymphocytes can't attack microbes unless they have permission. Killer T cells can't attack cancer cells unless they have permission. Why? Well, we're afraid of bacteria and cancer, but we are also afraid of white blood cells which act inappropriately, which cause immune reactions which they shouldn't, which start autoimmune diseases. So this cell's dangerous. It's a killer. It says, hey cell, could I see some ID? I don't like what I see, I'm going to kill you. That's dangerous. And it's possible that this cell makes a mistake. But if this cell wants to kill this body cell, but can't until it gets permission, this helper T cell is now involved. And now, even though it is possible that both make a mistake, that this one wants to kill this normal body cell and this one wants to kill a normal body cell, it's less likely. And so here's just a check that helps prevent autoimmune diseases. B lymphocytes need permission before they can just start making antibodies. Cytotoxic T lymphocytes, they need permission. And so T helper cells, in being required for permission, help to then prevent autoimmune disease. So once again, here's a B lymphocyte. It doesn't interact until it then gets permission, and the same with the cytotoxic T cell. This cytotoxic T cell can't kill the body cell. Look, it's sitting there doing nothing until this T helper lymphocyte comes along and gives it permission. Last step, and then we start to wind down. T helper cells need permission, all right? So T helper cells have to be activated before they can go around and activate others, okay? Who activates a T helper cell? And this now involves cells of the innate immune system, like macrophages and dendritic cells. So here's a macrophage. It eats a neutrophil that has bacteria in it. Now, remember that MHC proteins can grab a peptide from the interior of the cell. This macrophage in its interior now has something which is foreign. It has peptides which originated from the infecting bacteria. They bind cell ID badges, MHC proteins. And now this cell ID badge is going to wear a peptide from the infecting bacteria. Now this is what's called presenting. So this macrophage is essentially saying, hey, look what I just found. There's an infectious agent in the body. Here I'm presenting this. So here we are presenting part of a microbe that's infecting the body. Now, there's different kinds of MHC proteins. There's MHC1 and MHC2, and these cells, they express a different kind. So this doesn't look like a cancer cell, a cell ID badge with something abnormal stuck to it, because it's making a different MHC protein that these cells make. These cells are called antigen presenting cells, or APCs. So it eats a microbe, an antigen, it presents it, and it presents it to a T helper cell. When a T helper cell binds it, it says, hey, you have an ID badge. Hey, you have a peptide that you're presenting. Oh, I guess this is an infectious agent. The APC then also gives this T helper cell some interleukin, once again, a local hormone, which will activate it. And so now this T helper cell is activated. Macrophages are very important antigen presenting cells, but so are dendritic cells. Dendritic cells can look spiky, But immature dendritic cells can do something which is incredible. Here's an immature dendritic cell. It met a microbe in the periphery. It's presenting it. But before it matures, it actually then travels through lymphatic vessels to go to, say, the thymus or a lymph node. It then goes to the places where white blood cells are trained. So it has information about what's infecting the body. It's now going to go to, say, the thymus and present that. And now the white blood cells which are being trained with information about what is actually infecting the body. So dendritic cells which have the ability to travel through lymph and now go to a place where white blood cells are being trained, that's incredibly powerful. So antigen presenting cells, they present an antigen. So they'll eat an antigen, something foreign, and then they will break it down and then wear pieces of it, the foreign cell on their membrane and say, look, I'm presenting this. This is something that we need to mount an immune defense against. Thus, as a result of presenting to a T helper cell, they can activate a T helper cell. So here's a T helper cell. While they're interacting, this antigen presenting cell can give it a dose of interleukin. This is now an activated T helper cell. The activated T helper cell can now go then to B cells and activate those. So the APC, the antigen presenting cell, gave permission to the T helper cells. The T helper cells now give permission to B lymphocytes and T lymphocytes. So macrophages and dendritic cells, they are parts of the innate immune system. They are very important in the innate immune system. But here you can see in this process of presenting, they can eat antigens and present it. They then have important roles in the adaptive immune response in activating the T helper cells as well. So once again, T helper cells then go around and give permission. Here's a T helper cell, which was activated by an antigen presenting cell. Now this T helper cell is activating the cytotoxic T lymphocyte. So now it has permission to kill this cancer cell. So this is quite involved, obviously. It's even more involved because there's actually different kinds of macrophages, different kinds of dendritic cells. Now, for an introductory understanding of the immune system, this is, perhaps a deeper understanding of the material. But nevertheless, if going into the immune system is important, then that might be something that you would want to pursue later. A couple final points and then wrapping up. This is why HIV is so dangerous. HIV targets cells which have the CD4 receptor. So viruses only infect certain cells. Then HIV infects cells with the CD4 receptor. That includes T helper cells. So AIDS targets T helper cells. And thus, someone who has full-blown AIDS, the number of T helper cells in their blood, it goes down to 0. And the reason then that AIDS is so deadly is that AIDS now inactivates much of the immune system. The B lymphocytes, they can't make antibodies because they don't have permission, because all the permission givers are dead. The cytotoxic T cells, they can't attack cancer cells or virally infected cells because they don't have permission. All the permission givers are dead. And so HIV, in taking out the T helper cells, then means that the rest of the immune system doesn't have permission to act. So this is incredibly complicated. And that's why I have both like a cartoon version where you have, cells, attacking other cells in the midst of an immune battle. And then you hear like the killer T cell and the T helper cell, talking about their roles with accents, just explaining the same thing that I just did, but in more of an animation, perhaps a little sillier. But, if some of what I said, confused you, then here's the T helper cell saying, hey, cytotoxic T cell, I give you permission. And then once again, I have, superheroes, and so perhaps, them talking about each of their jobs that, helps as well. So our adaptive immune response, our acquired immune response, it gets better. It learns on what microbes are in the environment, and thus it learns what it has to fight against. And this is, what helps us live so long. Our immune cells, here are cancer cells, and our, immune, system can actually target those. Our adaptive immune response includes both the B lymphocytes, which make antibodies, and are in charge of the humoral response, that immunity which can is active with humors, with fluids, because they have antibodies in it. But then also the cell-mediated response, which involves T lymphocytes. There are different kinds of T lymphocytes, the Treg or T suppressor cells, which help the immune system mature and help prevent autoimmune diseases. The cytotoxic T lymphocytes, which can kill cancer cells and virally infected cells. And then the T helper cells, which give permission. So if this is a cytotoxic T cell which wants to kill this cancer cell, the T helper cell, notice it giving the thumbs up, it had to give permission before this one then, is capable of acting on the foreign cell. All right. Now I have all of these animations because this is complicated, and I think it's very common that students leave a lecture and say, there's all these cell types, I'm not quite sure. So maybe you want to watch these individual videos or watch some of these a second time. The immune system is arguably the second most complex part of our body after our nervous system. But they protect us because remember, we have stuff. We have carbohydrate and lipid and protein. And just as we eat stuff for lunch from other organisms, the world is full of things that wants our stuff. And it's these immune cells in our immune system which protects ourself, which stops other things which would like to take our stuff for their uses.